Surely, at this point, you've seen Imperial Yeast's amazing selection of high-quality brewing yeasts over at imperialyeast.com. But you might not be as familiar with Imperial Yeast's special order yeast bank. In addition to their tried-and-true core strain selection, Imperial Yeast has a number of ale or lager strains, Brettanomyces, Kveik, and other yeasts available for special order. If you're looking to match a yeast strain currently in production, or just looking for something new to play with in the brew house, reach out to Imperial Yeast customer service to see what they may have in store. Special order strains are subject to a 10-liter order minimum and require a 2 to 3 week propagation time, but they might just be what you're looking for your next brew. You can get more information about Imperial Yeast's special order strains by calling 503-477-5826 or emailing customerservice@imperialyeast.com. Welcome to the Brew Lab. I recently saw a list of the top 10 IPAs in the US, including Russian River's Pliny the Elder, Bell's Two-Hearted, Dogfish Head's 60-Minute IPA, and Treehouse Julius, all of which feature a large addition of dry hops, which got me wondering, how do they manage hop creep? I'm your host, Cade Job. Today in the lab, I'm speaking with my major advisor, Dr. Tom Shellhammer, about hop dextrin-reducing enzyme activity, the freshening power of hops, or also known more colloquially as hop creep. So Tom and Kaylin Kirkpatrick first published on hop creep back in 2018, and we're still publishing research on the subject, of course, with my most recent paper on how terroir influences hop creep, which you heard about last week. But since that first pub, if my math is correct, Tom has published around a half a dozen other papers on hop creep and has certainly become an industry expert on the topic. Now, it's been a while since I had last had him on the show, so I thought, hey, let's do a show on hop creep. He knows a thing or two about this. He probably knows with some of the issues that places like Bell's and Russian River have had issues with hop creep and what they've done about it. He also is pretty good with the science behind it, so he knows that, the factors that influence it. And so coming off the heels of my master's thesis presentation, I thought, hey, this would be a great part 2 of a 3-part series on hop creep. Of course, last week, the master thesis defense. This week, talk with Tom, the hop expert, about hop creep. And then next week, Jordan and I will talk about hop creep for home brewers. So I'll get into the interview with Tom here in a few minutes. But first, if you haven't yet signed up to become a patron, please consider doing so. By becoming a patron of Brewlosophy, you get awesome rewards like access to Brewlosophy contributor recipes that we've never published, new discounts each month to YakimaValleyHops.com, and for $3 a month, access to a live monthly Q&A session with a special guest from the brewing industry. Becoming a patron is easy. Once you've contributed at that $3 level or higher, you'll get access to the private Patreon Facebook page where all of the previous Q&A sessions are available to watch. So guests like Vinny Chilurzo from Russian River, John Palmer, the Brewlosopher himself, Marshall Schott, and many, many, many more. All the information that you need to get access to this awesome resource is available at patreon.com/Brewlosophy, so go check it out. Thank you to everyone that's left a rating or review of the show. If you haven't yet done so, please go to this podcast service of your choice, as long as it allows for ratings, and give us a rating or review of the show. We're over 100 ratings on Apple Podcasts, which is awesome, and I'd really appreciate it if we can get above 200 or more, or let's go. Who knows where the moon is? Take a second, let us know what you think of the show, and help others like you find us. All right, feedback this week is from listener Jean Peter. I think I said that right. Jens Peter, maybe? I'm not sure. He says, hey, Cade, I'm a home brewer from Switzerland and longtime listener and new Patreon, thank you, of Brewlosophy and your podcast, especially the Applying the Science series, which fits with my poor English skills. Now, I'm sure it helps with a lot of people as well, too. Anyway, he says, I've just listened to your latest episode with Brian and Philip, and I'm fascinated by the idea of the gas hopper. I have a unitank with a glycol chiller and enough kegs to give it a try. I'm looking for a small pump to circulate the gas in that pressurized system. Then I hope to be ready to go. I reached out to Dr. Philip Wheatstock, and he gave me some more tips as well. The plan is to brew a SNPA, and he says, I guess that's a single hop pale ale recipe. He says a keg 5 gallons and gas hop the remaining 5 gallons with Cascade. When everything comes out good, I'll send some bottles to Mr. Wheatstock, and I'm curious to hear his opinion. Thanks for all the content and to all of you. All right. Hey, thanks, John Peter. That's awesome feedback. I hope that this actually works for you. And yeah, if you are able to let us know what happens whenever you get that single hop pale ale in the keg. I'd love to see. And so thanks for listening, and thanks for listening all the way over in Switzerland. All right, after this break, I'll be back talking with Dr. Tom Shellhammer about hop creep. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Firm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com.
Introducing Zaxby's new chicken finger tacos. One with pico and creamy chipotle ranch, and the other with bacon and avocado ranch. Chicken expert since 1990. Taco expert since now. Woo, saucy! Zaxby's.
The mighty and resilient Merrimack River, carving through the communities of our great region. My name is Linda Lorden, proud president of Merrimack County Savings Bank. And like the river that serves as our namesake, we're a constant yet ever-changing presence. Because to us, it's bigger than banking. It's about powering communities and putting people first. It's about knowing where you came from and where you're going. That's Merrimack style. Visit us at themerrimack.com.
Way back in 1687, there's allegedly a comment in the Georgica Curiosa that states hops are given to re-fermentation when added to finished beer. Almost 400 years later, we colloquially refer to this term as hop creep. And joining me today in the lab to discuss hop creep is hop expert, Oregon State University professor, and my major advisor, Dr. Tom Shellhammer. Tom, welcome back to the lab.
Thanks. It's been kind of too long, I would say, huh? That was your first interview when you kicked this off the ground?
That's right. Yeah, you were the first one way back in 2021. And I mean, we see each other every day. So, we talk about all the stuff and we've certainly shared a lot and even have now published a paper together. But yeah, you were guest number 1. We talked about Dr. Scott LaFontaine's dry hop saturation work. And now, of course, it's episode 169. I mean, that's crazy.
Pretty amazing that you've been doing this like every week for like 3 years now. That's unbelievable.
Yeah, a couple of skips here and there, but it's crazy that it's just been this long for us to get back and do a podcast. We got to have you back on earlier than that next time, but
maybe a little more frequently, right?
Yeah, exactly, a little bit more frequently.
But kind of see what's going on inside the lab continually, so you know what kind of fun stuff's happening here. So
exactly, and I mean, there's so much research that's going on in the lab. I mean, we've got all of Ron Samia's work looking at yeast biotransformation. You've got Cecile's work, which will eventually be looking at hop aging. You've got Lindsay, who just graduated, looking at kilning. There's so much going on in the lab.
Hop and smoke work. Some hemp and cannabis work. So a lot of interesting things right now.
Exactly. Yeah, it's been super fun for me to be in the lab, and I've certainly learned a lot. And of course, just finished my master's thesis a little less than a month ago, or maybe right around a month ago.
Yeah, it was end of last month. Right?
Yeah, exactly. And of course, listeners, you heard last week the defense, the audio from my defense, where Tom and I investigated how terroir and maturity influence hop creep. And the thought for this episode is the start of a thesis begins with a literature review of research related to hop creep. So I thought it would be fun sort of to focus this episode on the state of hop creep. What do we know? What do we think we know? What questions remain? And of course, Tom is the hop expert, but I just did my defense on it. So I thought, hey, it might be fun if we did a show where Tom asks these questions of me, sort of like the defense part that happens when the doors close, right?
Maybe not quite like the defense, but sort of, right? So
way more friendly. Although my defense was very friendly. It was a good experience, which is great, and lucky for me. But some kind of peppering questions, and then I figured we'd kind of go through hop creep and what we know. But one of the questions I had to ask first, though, Tom, is like, hops have been added to beer for hundreds of years, but it seems like most of the major research around hop creep has really only occurred in the past 10 to 15 years.
Yeah, around this re-fermentation thing, then I would say in the last 8 years, right? So it was with stuff that like in 2018 when Kaylin first started publishing stuff on this in my lab, And I think people were looking at it a little bit before that, 2017, but yeah, clearly less than 10 years. And dry hopping has been one of these things that has sort of taken off in the last 10 years. And then as it took off, there was like these, unintended effects that people began to discover. I remember being at a CBC meeting, and it was in Nashville, and it was the one before this last one. It's actually, I think I have it. This was no, I guess it was 2018. That seems interesting. It was so. Anyway, the one of the questions came from the audience, like, do you experience diacetyl production when you dry hop beers? And we hadn't seen that because we're not a commercial brewery. We're doing everything on like small scale or pilot scale, but still. And that was kind of like a peek under the tent of this diacetyl bloom that can occur as a result of dry hopping. And it's interesting to hear that and see, okay, folks are having issues that were sort of unexpected.
Yeah, it's crazy, right? I mean, we think of like, okay, IPAs just add more hops, traditional American way. But then it comes with all of these other issues when you do that. There's other stuff that happens. And one of the main things that happens, of course, is additional aroma. But this side thing is hop creep, or as it has come to be known, hop creep. So, I want to talk about that, dig into it a little bit. But since it has been almost 3 years since I had you on the show, I figured we should start with a little bit of introduction. So we'll do, your intro first. Tom, you are, of course, the Nor'Wester Endowed Professor of Fermentation Science at Oregon State University. You oversee the brewing science research here at Oregon State. Numerous accolades with related to brewing science and hop research, published an incredible amount of articles, prolific researcher of hops. You've been researching hops now and publishing research for 2 decades. How do you keep finding new questions to research?
Oh, that's a good question. So part of what we just talked about, like the hearing questions come up at a conference like the CBC, where people are saying, "Hey, something like this happens. What do you think is going on?" So there's that element. I think staying really close to brewers and connected to brewers. The brewing industry is a pretty fraternal organization, anyway. I think it's unique in that regard. You go to a meeting, and it's not like people are sharing all their problems, but people are not reticent to talk about like issues that they're facing. While in other industries, people are pretty tight-lipped and you don't really know what's going on. People may collectively be struggling with the same issue, but not talking about it and not really knowing that they're all in the same boat. But brewers, I think, are less concerned about that, in part because I think there's just less secrets in beer. Although I would say there's the same thing, there'd be less secrets in wine as well. We've been making beer and wine for millennia. And so, like, even though we get new science and we get deeper understanding of things, the problems that we're facing, I feel like everything that is old is new again. So staying connected with brewers is important. And then I would say my career sort of evolved maybe 10 years ago to start working more with hop growers. And part of this is coming out of work that is funded for the Hop Research Council. So initially, my focus was very much on beer. So like the impact of, I would say the first few research projects were trying to poke my finger at the conventional wisdom. Like how and why does this conventional wisdom appear, and is it real? Like brewers, and maybe other industries have this as well, but certainly brewers can be pretty dogmatic. They kind of have an idea of what something is, and everybody knows that that's just the way it is, and it gets propagated. And so one of the first ones of this was this concept of high cohumulone, and in beer would be high isocohumulone, but high cohumulone hops. And Lloyd Rigby, who was a hop researcher back in the, say, 50s, 60s, and 70s, or maybe 60s, 70s, and 80s, had come up with this idea, this theory that because of the weak acid nature of hop acids, that the qualitative differences among the individual analogs, like cohumulone versus humulone versus adihumulone, and then their isomerized components, behave differently from a bitterness perspective. And people like just bid on that hook, line, and sinker. And for years, people, breeders and brewers are looking for, it's got to be low cohumulon hops. This is like one of the things that breeders are always trying to achieve. And it's difficult. Just naturally, when you breed hops, you see a higher proportion of cohumulon. So I was thinking, like, this seems like a really subtle effect. Why would we be so like focused on this? And so I started poking my finger harder and harder on that. And then came to realize through several years of work that was funded by Hop Research Council and also of Johnny Haas, that isocohumulon doesn't mean anything. And if anything, high isocohumulon hops or beers that are higher on isocohumulon tend to be, the bitterness cleans up quicker, the bitter quality is smoother, less medicinal, but it's really, really subtle. That's why I first came up with this. I came up with this, called this term like snowflake tasting, because we're just tasting these beers that are all the same level of bitterness, but they have just different ratios of cohumulon, isocohumulon, and isohumulon, and it's just really hard to get a sense on whether there's a difference or not. So, so anyway, it was like initially just poking my finger at like things that is whatever the conventional wisdom is, to see, is there really some there in that regard. And then during that, so that was like focusing very much on beer and beer quality and the effect of hops on beer quality and bitterness quality, beer flavor stability, hoppy aroma in beer. That was kind of stuff that Daniel Sharp and Dan Vollmer and then Scott LaFontaine were working on. And it was about at that point where I could see, and actually some folks within the HRC said, hey, the folks on the hop side could use like some, not help, but like there are problems there that are valuable for brewers and also for hop brewers. So then we started working our way kind of up the value chain, so to speak, that we started looking at what is it in hops, and this is kind of where Scott LaFontaine was working on, what is it in hops that really drive hop aroma in beer, and what kind of things affect that. So maturity has a big influence on that. And certainly, obviously, hop variety does as well, and trying to understand what components in hops are leading to these, the intensity of hoppiness in hops and in beer. And then next came kilning work, like how does kilning impact hop quality? And then with that, as at about that same time, we were starting to do this work on hop creep, and we saw in some of Lindsay's work, okay, maybe there's like a rep effect that would lead to questions around maturity and hop creep. And so we kind of revisited the hop maturity question, but this time with the focus on this hop creep. And so it's like, these dialogues with researchers, and you kind of saw it yourself when we were at the HSC meeting last week, which was here in Corvallis, that you have a lot of stuff, a lot of this talk happens when you're on a bus going on tour or having a beer with somebody, when you start talking about what they're interested in. And so in doing that, you kind of hear what are some of the problems. And there's some instances where people actually come to us and say, hey, this is something that we think is like an issue. Can you help us solve that? But yeah, it's just dialogue with brewers and hop growers, and just being, I wouldn't say data driven, but just being open and listening to what's going on, and not trying to come at it with preconceived ideas. Because I found that like not craft brewers, home brewers often have like insight that brewers may or may not have, because they're so into like their home brewing, and many of them are really nerdy. Many of them are scientists that are doing this. And so it's like, when you hear that, it's not that it entirely drives my research, but it informs my research direction.
Right. Questions can come from anywhere, right? And they come from all over the place. They come from home brewers. They come from conversations at HRC. They come from CBC, right? Comments during a question. Hey, have you experienced diacetyl? I don't know. Is there something there? And then little did you know that was going to spawn like 6 or 7 years worth of work.
Yeah, I know, on this hop creep. And we should talk a little bit about hop creep, because like the hop creep and the name hop creep, that's another CBC thing. We were introducing this topic, and it was Q&A session afterwards, and somebody had the mic and they said, yeah, in our brewery we call it hop creep, kind of this creeping slow change in final gravity post dry hopping. And it kind of makes sense. I can understand that, like the the what it's conveying is like this slow, very creeping, almost imperceptible change in final gravity. But I don't know, it's not necessarily the best word. I don't know if I have a replacement because like the
Well, there is that replacement.
fermentation or
Yeah,
re-fermentation or
or the freshening power, right? That was one that I was going to ask a question about a little later, but that was the one that the folks at Bell's liked.
yeah.
That Luke Chadwick and Jake Kirkendall, who now of course is at CalSec. Again, working with one of your former students, also at CalStech. They kind of coined this term that Brown and Morris did in 1893, which was the freshening power of hops, right? When you add hops, it causes this re-fermentation that sort of scavenges any maybe remaining oxygen that might have been in a cask, right? Because hops were getting put into cask.
Yeah.
It drives attenuation, dryness, all this stuff. Yeah.
Yeah, it certainly would drive carbonation. It would make the beers more lively, as the Brits would say back then. It probably would say still now. Yeah, I don't know if freshening power conveys the phenomenon. I don't think brewers view this as necessarily a positive element to their brewing practice. It creates headaches, right? It creates headaches in terms of delaying the time at which the beer moves through the brewery because of either this slow time to attenuation, or more often the fact that you get this big diacetyl, not big, but you get a diacetyl bump, and then it just takes forever to get rid of that. And so, beers are out of spec and they need to be blended and they're moving slow. And the connection, even though I could understand maybe Chad's idea of like from a British centric perspective of what you just talked about, scavenging oxygen and adding new like carbonation to beers that were inherently low in carbonation to begin with, these cask ales, that it could be seen kind of like a freshening, but yeah, it doesn't that term doesn't like resonate with me.
It's interesting, right? Because we spend so much time looking at those problems. Like you said, like there's this diacetyl creep that we see oftentimes, or, I keep using the word creep, but, there's this diacetyl of flux. There's an increase that happens right after you drop the hops in, and some yeasts. Some yeasts it doesn't, some brands it doesn't, but often you see this diacetyl increase, and then, it just sticks around forever. And even Luke Chadwick at Bell's, when he was looking at like, hey, can we use hop creep as a carbonation mechanism? Can I put like hops into bottles and then make that carbonate bottles? Right. Yeah, and he just created a whole bunch of butter bombs.
Yeah, when you create butter bombs and you don't really know where the end point is.
Yeah.
When you bottle condition beers and you put a fixed amount of fermentables in, then you know what you're going to get when it's done, right? Those fermentables get chewed up, they get converted into ethanol and CO2, and you know what you got. In this case, you don't know the extent to which those residual dextrins that are in the beer post normal fermentation, pre-dry hop, are actually going to be converted into fermentables. And it's slow, man. It's really slow, but it occurs. It occurs like you measure commercial beers that have been dry hopped that are old, you'll find high levels of glucose and maltose, not high, but higher than what you would expect at the end of fermentation. So
yeah. That is, of course, right. If you don't, if you package them, but then don't, filter them or something, or pasteurize and get rid of the yeast and stuff.
I would say even if you don't pasteurize, like if you have a beer that's been centrifuged and the cell count going into that beer is really low, yeah, you have the possibility for that re-fermentation to occur. Where the problems have occurred is where folks were bottle conditioning beers. So like when the folks at Allagash, when they kind of bumped into this, because they do bottle conditioning on all their beers and or at least most of the beers, and so they pay very close attention to bottle repressurization because they need that in order to release their packages to the trade, and saw some serious overpressurizations. And not many people do, like if you look across the board, do bottle re-fermentations in the U.S. And so that's the combination there. Like if you dry hop and then do bottle re-fermentation shortly thereafter, I guess it depends on how shortly. It just depends back to how much substrate do you have? Like if some of those dextrins are being consumed in the cellar prior to the sugar add back, as a priming for bottle re-fermentation. If that's occurring in the cellar, you're not going to see as big a bump. But if it hasn't, and it happens in the package, then whoa, then you have a problem. And it's
Yeah,
I don't
exactly.
understand.
Yeah, especially when you're bottle when you're using a bottle conditioning, right? Because like you said, I mean, we're also assuming that the wort or the beer that's in the fermenter is the same all the way through, right? That the dextrin profile is exactly the same. The concentration of those dextrins are the same throughout. There's going to be a little bit of variation and
there'll be a variation from crop year to crop year. There'll be variation and
from batch to batch,
mashing regimes, right? Exactly. So that's would be the challenging part of trying to use hop creep as like a re-fermentation tool.
Mm-hmm. And so that kind of, brings up again, like we talk about some of the challenges that are associated with hop creep. And I think, of course, Brown and Morris and also Jake Kirkendall and Luke Chadwick looking at the freshening power, kind of looking at this from hey, if you can avoid the diacetyl, maybe this actually helps out some of those British beers that were in casks. So I can certainly see that. But yeah, there's sort of like, I guess before we get into it any further, we should really quick back up and just make sure everybody's on the same page about how hop creep works and like what it is, what it does. And so hop creep is that we call it dextrin reducing enzyme activity, right? It's the dextrins that are present in beer that are the result of the way that we mash, right? It's the result of using our mashing.
It's the way we mash, and the fact that the substrate we mash, that is malted barley, it has an enzyme package that's created during malting that will break down the linear portions of starch, but has very little in terms of the ability to break down branched starch molecules. And so what happens at the end of mashing is that you've got this large pool of fermentable sugars, mainly maltose, but some glucose, and then some maltotriose, and which can be fermentable depending upon the yeast you're using. But then you've got this big pool of branched dextrins of different sizes, where the enzymes that are tearing down the starch during the mashing process no longer become effective because they can't deal with the branched nature of the starch polymer. And that kind of gets into what, starch chemistry and whatnot. But in the simplest sense, you got a mixture of lots of straight chain polymers, and some of these have branch points in them. And the enzymes that the barley produces can break down the linear portions, but are fairly ineffective at the branched portions. And so at the end of the mashing process, you've got this pool of, you start with big starch molecules, and at the end, you're left with this pool of really small molecules. The really small ones are fermentables. The medium small ones are not fermentable. And that non-fermentable portion, which we're just going to loosely call dextrins, or if you're a brewer, it would be called like the residual extract, which is not all dextrins, it's protein and minerals and other stuff. But that portion of that unfermentable portion that is left behind can be as much as like 25 or 30% of what you started with, and from a soluble material, like when you make wort, the potential that you could get out of that in terms of all the extract, you could convert, let's say 70% of that extract into fermentables, but 30%, 25 to 30% doesn't get touched. It gets passed on through the finished beer. And that's what part of the calorific content of beer, the carbs are in there. The difference between, let's say, Bud and Bud Light is the degree of fermentability. The Bud Light will have less of these residual dextrins. And so brewers can add exogenous enzymes or do really long mashing regimes to try to break those enzymes, the dextrins down, and make the wort more fermentable, which lowers the amount of carbs that get carried through from the mashing process through the fermentation process into the finished beer.
Mm-hmm. Yeah, and I would guess at that point too, like if you make a really fermentable mash, then you also don't see as much creep.
Exactly right, because there's no the right. So we're setting the stage like it's the unfermentable portion of the wort that is the starting point for this hop creep. There are enzymes in the hops that can attack those dextrins. So there's some of the same enzymes that you find in mash, in barley, but there's also some there, very low levels, that can break down those branched dextrins. And so you start seeing these, what would you consider to be non-fermentable dextrins become fermentable because the their size is broken down to the point that they're small enough that yeast can consume them. But it's slow. It's a slow process, um, and as you pointed out, if you've got more of those dextrins, you're going to see more of an effect. If you got less of those dextrins, like with a light beer, you're going to make like a brewed IPA, you're actually adding exogenous enzymes to that to try to dry that beer out. And it's not going to have any hop creep effect because there's just nothing there for it to work on. And so some people might say, "Oh, that's the solution." It is if like you want a world of just really dry beers, but so.
Sure. Yeah. It's that unfermentable portion, that residual extract that's sitting there at the end of the beer. That's certainly like the starting place for where what gets liberated and why we see the effects of hop creep, like the attenuation and the alcohol, CO2, diacetyl, all that sort of stuff. But we call it hop creep, right? And so one of the questions that has really been popular over, especially like the past few years. I mean, I'm talking like even as recently as like 2022, 2023. That sort of stuff, is where are these enzymes coming from, right? And so we call it hop creep, I think largely because when you add dry hops, you see a creep, but are the enzymes actually coming from hops?
Yeah, that's a really good question, one that we haven't fully answered yet. Like whenever I present this to a scientific audience, I always refer to these enzymes that are associated with hops. They are either in hops or on hops. Leaving open the possibility that the enzymes could be of a non-plant origin, that they could be associated with some fungal or bacterial species that are
right.
hops.
Right, kind of two ways to think about it. They're either the enzymes are either present in the hops themselves, or they're the result of microbes that are existing on the hops, right? Because hops don't go the only kill step, quote unquote, the only like neutralization step that they go through is potentially kilning. And we'll see some results in the next segment about how kilning temperature impacts hop creep potential. But microbes, themselves are present. And so either they're present on the hops, and when they get put into this starch solution, they liberate the residual dextrins, or the enzymes are excreted out of the hops, or the enzymes are actually coming from the hops themselves. And it's been sort of a little debate, a little scientific debate.
Yeah, I think it's been like a scientific debate. Practically, it doesn't really in the short term matter because the hops have enzymes on them, and the brewers are trying to figure out, okay, how do we work with these?
Exactly. What do you do?
but it is kind of interesting if you understand the source, then you have a way of actually kind of tackling the problem rather than just. It's kind of like you have a headache, so you take ibuprofen. You don't really understand what the issue is. You're just kind of treating the symptoms. It's kind of like that in this case. So is it like microbiota derived or is it plant derived? And I would say after talking to a number of different folks, plant scientists, plant breeders, microbiologists, that there's like a preponderance of evidence that points to the fact that they're in the plant themselves. And for a number of reasons, like you just don't find organisms excreting huge amounts of dextrin reducing enzymes on a plant that doesn't have much starch to begin with. In terms of like a pest host interaction or organism host interaction. And even if there are organisms that would do that, and there are, like if you culture up what's on hops, you'll find some bacteria and some yeast. You're not going to find a lot. It's not like these plants are just full of microbiota. They're not sterile, but they're not just brimming with microflora.
Yeah, I mean they go through a kilning step that kills off.
Yeah, exactly. That kind of has a pasteurization effect on it as well.
Yeah.
Talking to microbiologists, they're like, if these organisms are actually living, reproducing, metabolizing carbon to provide energy, then you might see an effect. But even then, most organisms this is all happening within the organism itself, as opposed to yeast or fungi excreting lots of enzymes outside of the cell to try to attack things. Now, there are some organisms that can break down, like fungi that can slowly break down wood. They excrete cellulases, and actually we manipulate that when we look at trying to create different enzymes will take different fungal species like Aspergillus or Rhizopus and do some GM modifications of them because they naturally produce high levels of enzymes. But it's not like when you have a fungal attack or outbreak in a forest that the trees all of a sudden come melting away because of all this enzyme excretion. So the possibility that there would be really high levels of enzymes, not high, but high enough levels of enzymes sitting on plants that would have this effect is less than the fact that the enzymes are probably coming from the plants. And we talked to, like, during your defense, we had two plant breeders on there. We had a barley breeder and barley geneticist, and we had a hop breeder and a hop geneticist. And their comment was like, okay, everything in your defense on this terroir talk points to the fact that the only real big effect, not the only, I would say the largest effect that you saw in your results was hop variety. And so once a variety starts trumping other influences, then the plant breeders say, okay, this is like an inherent trait within the plant itself. Like if you can see differences between two different subspecies or strains within a species, then okay, there's something that's going on with that organism itself. There's not something outside that. And we've seen that, like the work that you published on the terroir effect, or let's call it maybe in effect, is in some respects replicating what Arnie Stockholm was publishing on when we did kind of the run-up smaller scale terroir study ahead of what you did, which was that, okay, there's some variation between the fields, and there's actually a decent amount of variation within the fields, and those, create their own interesting questions. But the big takeaway was that the hops, despite this variation within fields and across fields, the hops were different based upon variety. Variety seems to be the biggest player. And that is reinforced by what brewers tell us. When you talk to brewers, like, oh yeah, we know certain varieties tend to be more prone to producing hop creep than other varieties. And so that effect, and there was a little piece in like Arnie's work where we actually looked at disease pressure as measured by the amount of fungicide applications that the growers had to spray to keep this disease in check. And so the greater the frequency of sprays means the greater the fire. They're having a battle this fire. And so it means that the greater the fungal pressure. And there was like a really strong, for Simcoe, a really strong linear response that was inversely related to disease pressure. That meant the greater the fungal issue that was going on in the field, the less hop creep. Just like, okay, that kind of runs almost counter to this idea that the microbiota is having an impact. Now, given all that, we do see that you saw differences within like regions. So like within Oregon, we could see pretty broad ranges across the different fields. And we see that like in your maturity study that we look at like hop creep that is occurring across 3 different fields that we've looked at. This is that YCR. I forget, there was like field 7, field 20, something or other. You look at those year on year on year, they're all different. They're not hugely different, but they're different. Each field is different. It's not like the data overlap. So there's something going on within the field, and whether that's plant health or something around the agronomics, it's hard to say, but
the variation, right? That like the plants grow differently. I mean, we're looking at 3 binds, in that maturity study that's grown in a field.
Yeah, right, exactly. So
who knows what the variation even is? Even though the fields seem to be different, there's field to field variation and within field variation too. But that just also speaks to the plant itself, right? Each plant is unique, even though it comes from the same DNA. It's growing differently. It's got different
somewhat unique.
pressures.
That's one thing to keep in mind, that the plants in a field of hops are clones. Right?
That's right.
So it's the same thing. It would be like apples and grapes, that these are not sexually propagated plants. They're clonally propagated. So, kind of in theory, when you look at a cascade plant, they're all identical to the one original cascade plant, and they all can be derived from that. Now, that doesn't mean that they're all propagated from that one original cascade plant, but because they're clonally propagated, it's like, whatever, like taking a lizard, pulling off its tail, it grows another tail, right? It's coming from, and if you could grow a lizard from the tail, then you that would be more like a different kind of clonal propagation. But they're all genetically identical, and but then they respond differently
To the environment, right?
the environment, right?
They
Exactly.
grow
You've
differently. Yeah.
got the soil characteristics are different from field to field. There's a lot of stuff there in terms of nutrients. Like there's a question around like back to the host microorganism interaction of like the role of the microbiota in the soil and how it influences nutrient uptake. So those are going to be different from field to field. And so there are differences.
Yeah, I mean, and each of those pressures, right? The soil, the sunlight availability. I mean, any of these things, each of these pressures treat the plant differently, and they cause different like hormonal responses, and even different gene expression, right? Of the hops have the same DNA, but the genes that they're using or looking to that DNA code to use maybe different. Such that there is field to field variation or even like bind to bind variation, but if the genome is roughly the same. And wildly different, than say something else, say like, Citra versus Cascade, maybe that it does sort of still lend itself to an explanation under the, the DNA root.
The genetic differences of the plants themselves, they're not wildly different, but they're different enough because the phenotypic expression of these plants produce hops that, smell like Cascade or, create Cascade versus Citra versus Mosaic. And they're all hops, but they have different, say, vigor. We were in a field last week looking at Citra and Mosaic, and like they're just really different looking plants. They all look like hops, but like Mosaic are these big bushy plants, lots of cones, and then Citra, also big plants, but not as bushy and more leaf area than cones, and of course they get different aromas and they mature at different times. And
yeah. And I bring up the DNA piece of this too, because that seems like that's what's kind of re-kicked off or maybe revitalized this whole, debate. I call it a debate. I mean, I don't know that it's necessarily a debate. There's certainly, room for more exploration, but it's the Matt Cottrell's work from 2022 when he discovered, hops or the hop. I'm sorry, when he discovered hop creep or amylase DNA. There we go. I can finally get my words right.
Yeah.
He discovered amylase DNA that's present in hops. So hops have at least some of the ability to generate amylases.
Yeah. I don't know how I would
themselves.
call it discovered, because that's a true a little more to this.
True.
We know that the plants have these enzymes in them, and so he was kind of confirming, okay, I can see genes that would express these amylase enzymes. And it was kind of neat because he could look at those, and he could also look in cannabis, and then he found some proteomic work in cannabis that sort of demonstrate the expression of those genes and the actual production of those enzymes. And we don't have that data on hops or humulus, so it was a nice way to kind of like say, okay, we can see the genes in cannabis, we can see the expression of these enzymes in cannabis. We don't know if they're expressed in hops. We can see the same genes in hops, but they perform very similar. Like if you dry hop with cannabis and dry hop with humulus, you get the same kind of hop creep response. And if you take a botanical plant, I think you took oregano, it doesn't have this. Okay, you don't get it, right? Yeah. So
exactly.
I thought that was a pretty cool approach.
Yeah.
I'm more about arguing around the semantics of it. It's not like
No, of course.
Amylases exist in plants. I think we've known that for
yeah. Well, but you make that point, but it's like it seems like that's where the conversation was for brewers for a long time. Is like, okay, yeah, like there's no way hops aren't starch producers, right? Barley's a starch producer.
I remember the first time somebody kind of just looked at me and like I was like an idiot almost, because they're like, well, of course plants do this. He was like, Tom, how in the hell do you think these plants survive at night? Right? Like this is the basic plant biochemistry that plants photosynthesize, they make glucose, that's how they survive. That's why plants are green. Okay, I understand that from like high school biology. That's what chlorophyll does. And if you have excess sugars that are produced, it's stored as starch. And then at night, when there's no sun, it's like the plants all go to sleep and just all of a sudden, like you've just turned the electricity off and there's nothing going on. And take that starch and break it down and continue, living. I was like, I guess you're right. Yeah, plants need something. They need to be able to survive overnight. So
yeah, And so yeah, it's like you said, it's not like they discovered it. Maybe a better way to say this is just it's the first time that the connection was explicitly stated to brewers,
exactly.
or as
Right.
related to hop creep, right? Like that this is clearly how plants metabolize at night, and so yeah, here's the DNA evidence that you need, brewers, and
Exactly.
like you said, it's a really cool novel experiment too, looking at like, oh, if we dry hop with cannabis, I can see hop creep, but if I dry hop with oregano, I don't.
Yeah. One of the things we did when Kaylin was working on this project, which was really fun, was she went out to the Louisville farm and picked hops at like 2 p.m. when the sun was high in the sky, and then she slept out there and picked hops at 2 a.m. when the plants should be like, it's been dark for quite a while. And we brought those back to lab. We were trying to look at, okay, if there is this difference, because this is kind of provoked by this conversation that I had in a hop field with a plant scientist, like, okay, maybe there's a real like diurnal difference between daytime hops and nighttime hops. And that kind of, I was wondering that maybe that's a reason why there's so much variation in some hops and hop fields, like maybe time of picking, because hops aren't just picked in the daylight hours, they're picked, around the clock. But the data that we got from that was just kind of like unsatisfying. There was dealing with wet hops and trying to do the assays with them, and it's like, but it's something we should look again.
Yeah.
And maybe that's something that you're going to spend a lot of time in the hop field at odd hours. So maybe that's what you need to do.
Right. Or go grab some out of the kiln and be like, okay, here's the ones that are out of the kiln at 3 a.m, and here's the ones that are coming out of the kiln.
Well, we could do that.
But also,
like, when you show up for these smoking trials at 5:30 in the morning, when it's still dark, like that might be a great time to sample a hop plant and see, okay, let's see what the enzyme activity is.
Sure. Yeah, it's interesting, right? So, I mean, even just looking at, what is hop creep and what are the sources of hop creep, you and I've managed to talk about that for over half an hour, right? And
I know, exactly.
And find, and still find room for additional research, which I think is really cool. That kind of answers that question I asked you earlier. How do you keep finding questions? It's having conversations like these, which you and I have.
This is kind of like what science is about, right? I think I feel lucky that I get to work on like hops and beer, and we have these conversations about. I think are really cool.
And then we get to have conversations like this too. It's fun. I mean, being part of the lab, we get to have conversations like this all the time. But unfortunately, we do have to take a quick break from the conversation, pay some love to sponsors. And then when we come back, we'll talk about methods for measuring hop creep and also some more about those factors that influence hop creep. We'll be right back. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brewlosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app.
Established in 1995, More Beer has been consistently serving the greater brewing community since the times IPA was expected to be bitter and clear, and there are reasons they've stuck around so long. In addition to their massive product selection and excellent customer service, More Beer has locations on both the west and the east coast of the United States, which translates into fast shipping times regardless of where you live. And when you spend more than $59, shipping is free. When you're in need of brewing ingredients and gear, there's no better option than morebeer.com, one of the most trusted shops on the planet.
The Labor Day weekend sale is on at Tanger Outlets. Score up to 70% off at top brands like Nike Factory Store, Crocs, J. Crew Factory, Gap Outlet, and so many more. Refresh your wardrobe with the latest arrivals for less from all your favorite brands this Labor Day weekend. Tanger Outlets, always on trend, always on sale. Plan your trip at Tanger.com.
Introducing Zaxby's new chicken finger tacos. One with pico and creamy chipotle ranch, and the other with bacon and avocado ranch. Chicken expert since 1990. Taco expert since now. Woo, saucy! Zaxby's.
Brewers take so much time and effort to craft shelf-stable beers that taste great when they reach consumers, but if they're not paying close attention when making those beers that have a dry hop addition, hop creep can cause package instability, increase in alcohol, attenuation, and a host of other problems that might actually reduce the shelf stability of that specially crafted beer. So, Tom, one of the things I find fascinating is that a story you had told me, and even as recently as last year, it's that sometimes I often hear brewers saying hop creep doesn't exist, or that's not a problem. We don't have that problem. I mean, even at a presentation that you and I were at, about 2 years ago, I was presenting on the terroir, and one of the people at the Q&A discussion at the end said, "Why are we even still talking about hop creep? It's not a thing."
I remember being confronted with the same question. I think it might have been like Garrett Oliver at one of the CBCs in that same Q&A session. It wasn't the one with the diacetyl, but it was where someone had said, "Yeah, this week in our brew we call this hop creep." And he asked me, but also asked the crowd. He's like, "If this is really happening, why aren't bottles of beer just blowing up on the shelves every day all over the country?" And I was kind of the same. Yeah, I was like, I don't know. I was at a loss, and
I don't know. I haven't studied this yet.
Yeah, and so. So I think, it was shortly after that we worked closely with a handful of brewers, a handful of commercial brewers that your listeners would know and would be eager to drink their beer. We had 4 or 5 different breweries from different parts of the country who were sending us beers as they were dry hopping beers, kind of pre-dry hop, post-dry hop, post-packaging. And we were tracking them. And it was interesting to see that 3-4 months after packaging, you didn't necessarily see the package pressures changing because the beers had been filtered or they'd been centrifuged. They weren't pasteurized, but there just wasn't enough yeast, and the conditions at the end of fermentation are presumably harsh enough that having a few cells of Saccharomyces in your beer is not going to just make it suddenly re-ferment. Like you need a decent amount of yeast. But what we could see in all these beers was that the enzyme activity was continuing to occur. That is, the ratio of, the extract itself, residual extract, was staying relatively constant, but the makeup of that was changing. Like beers, when they're done fermenting, should have really low levels of fermentables, right? Because the yeast should have mopped things up. It's not always the case. You can find lager beers that have still residual fermentables. There's some yeast that are just really, like lager yeast, that are just really poor attenuators that will leave a little bit of residual sugar left behind. But what we see then is that those dextrins start getting reduced, and the amount of glucose and maltose starts increasing. And part of it was wondering, maybe this beers actually gets sweeter over time, but maltose surprisingly doesn't have a really high sweet intensity. You need actually a fair amount of maltose in beer. We had a student basically start doing maltose additions to beer, and we would just taste them in the lab. And you need to get quite a bit of maltose in there before you would start noticing that they actually start tasting sweet. But that doesn't mean that biochemical changes are continuing to occur. So I think the hop creep is occurring, and I would say that if you're dry hopping beers, it's happening. It's just like the extent to which it's happening, and that's why I think it was hiding in plain sight for so many years. When we first started doing this work, one of my former students who was working at a very large brewery, not very large, but a large brewery, I asked him about that we were working on this topic, and that it was really interesting to see the amount of re-fermentation that occurs and in the finished beers. And so we talked about like final gravities of the beers, and so we have these beers that they start at a final gravity of like 3.5, and they start dropping to 2.5, and he's like, "What do you mean? Like we never see like beers at 3.5. That's kind of you're something must be wrong with your brewing process, because they all land at 2.5 or 2, 3, I don't know, whatever it was for him." And that's because like their flagship beer was a dry hop beer, and it was dry hopped for like 3 or 4 weeks. And so it was always creeping. It was just, they spent plenty of time in the cellar. They hadn't actually really checked the pre-dry hop attenuation level, because you're dry hopping right out of fermentation. It doesn't make sense to track that. So it was occurring, but they didn't realize it was occurring.
Yeah.
Well, what we discovered with some of Lindsay's work on the kilning was that you got these different like let's say with field-to-field effects, or maybe it's a maturity effect, where the level of the level of hop creep is just inherently different between two lots of the same hop variety. And what we could find is that the degree of that hop creep potential had an influence on whether you actually saw the effect in the finished beer. Like we could see the effect of kilning as we increase kiln temperatures. We could reduce the amount of hop enzymes that are present in the hops. But then we would take those hops and we would actually brew with them and dry hop with them. And in some cases, we would see this creeping phenomenon, but it was really subtle. And in other cases, it was really huge. And what we noticed is that there's like a threshold, right? Like once you get down below a certain level of enzyme activity, the effect that you're going to see in the brewery is low. It's still occurring, but it's low-ish. And it's problematic. Then it would be problematic if you were to do like a bottle conditioning of those beers, because you've added yeast and now you have this trickling in of more fermentables. But for hops that have a really high amount of this potential, it's like really observable. And some people are seeing, like almost a whole ABV jump after dry hopping, while others see very little, right? So
and like we said, like diacetyl and all that sort of stuff. It's sort of like, it's like you mentioned earlier, right? The theme through all of this is that we use ibuprofen to treat the cold, right? But ibuprofen doesn't stop the virus or the bacteria or whatever it is that caused the cold in the first place. It's just a symptom.
Effect, right?
Yeah. And so and you made that point, right? That like some breweries weren't seeing it. Well, yeah, because their brewing practices are a little bit different, right? I mean, they're used to all of their beers finish at 2.5. So they're waiting until those beers finish at 2.5. They're not measuring the gravity. Well, they may be measuring the gravity, but they don't care what the gravity is before they dry hop, because they're going to dry hop.
It's still, right? Still like they're in the progress of their fermentation.
Yeah.
Right.
Or maybe it's like what most brewers do now, which is like you're dry hopping at or near the end of fermentation. So if you know you're going to finish at 2.5, you dry hop somewhere at like 3.5 or 4. Right. So you're just like a degree or two towards the end of fermentation.
And if a beer has been designed to be dry hopped from the very beginning, you don't have a non-dry hop counterpart as like a brand to compare to. So
the other big symptom treatment that people use all the time is ALDC, right? The alpha, I'm going to say it wrong, alpha-ketolactate decarboxylase. No, I got it right.
Yep.
Yeah. That's also the treatment for the symptom, right?
Yeah,
I mean, the symptoms.
Yeah, and that ALDC treatment to try to minimize diacetyl is basically a way of mitigating large diacetyl spikes is by adding this enzyme that will actually convert the alpha-ketolactate, which is the precursor to diacetyl production, kind of bypasses that diacetyl production, which is a pretty clever approach. And so brewers will use that. It's not like a guarantee to prevent a big diacetyl spike, but it will certainly reduce it. It's kind of like the ibuprofen thing. Like if you got a full-on migraine, ibuprofen's not gonna do it. You can attack it before it gets really bad. You're probably in better shape.
Yeah.
So,
exactly.
But talking to brewers, like when I asked, like in a large conference, like at a CBC, there might be kind of lectures over like 1,000 brewers sitting in this room, and I asked people to raise their hands if they experience hop creep, and I would say like 75% of the hands go up. And then I ask, okay, how many people have diacetyl problems as a result of dry hop creep? And only a quarter of the hands are up. So it's a problem that it occurs, and when it does occur, it can be way worse than just the fact that you've got beer that's over attenuated and it's taking a little bit longer to finish. So I think there's something there, and that would be interesting to study. It's something I think James Bruner was trying to tackle in his work. But we had a student here getting ready. Arnie actually was starting in on that project, and it's a pretty complex project to work on, because you need to get a beer that you're going to dry hop that could throw some diacetyl, and then try to play around with, if you have different yeasts. And so it could be attacked a number of different ways to try to answer that question, but it's not as easy as just like, oh, we've got hops, we put them into wort and we just measure extent of attenuation.
Well, so that was going to be my question, right? Because if we're trying to it's kind of like, trying to address the symptoms, then how do you measure the hop creep? As you mentioned earlier, right? You might have some of these, you had a lot of these bottles that were getting sent to you that because of the practices of the brewer, there wasn't enough yeast in the bottle. So you don't see what we would say, quote, the typical, hop creep, effects, right? That over attenuation, that decrease in residual extract, the increase in alcohol.
Overcarbonation or high levels of diacetyl, because there wasn't any of the yeast biochemistry going on. What was going on was the enzyme enzyme biochemistry that was still going on, unless the brewers pasteurized. We had a brewer that sent us one of the IPAs, and they have a pasteurizer and they pasteurize their beers, and that solves the problem.
Right, you denature the enzymes.
Those beers did not change, and I think there's maybe we may be moving into a phase where brewers, I don't know about consumers, but brewers will be a little bit more open to the idea of pasteurizing beers as we start seeing more and more NA beer production, because you cannot homebrew your way into an NA beer production.
That's a tough one. That's a scary one for sure. So much sugar and dextrin that's available there. If you get any kind of microorganism in that, I mean, it's just going to have a field
exactly.
day.
So I think that maybe brewers will open up to the benefits of pasteurization, and I think there's some tremendous benefits from a flavor stability, because the enzyme problem that we've been studying now for 7 or 8 years has been just one enzyme or a class of enzymes that break down dextrins. But I'm sure there's a whole suite of other enzymes that the hops are providing. There could be enzymes that are oxidative in nature, and those would not be good from a flavor stability perspective. And pasteurization of beer could be a really good thing in terms of stabilizing other biochemical derived changes that may be occurring either from residual enzymes that are left behind by the yeast post fermentation, or because of high levels of dry hopping.
Yeah, exactly. So if I am a brewer and I'm curious and I want to know if I have a dry hopping, or if I'm seeing diacetyl in my beers or something like that, how do I go about measuring it? What are some of the methods for measuring hop creep? What do I do?
Yeah, the easiest way, if you're curious to see this as a home brewer, and you can measure this with a hydrometer, right? You just need to be fairly meticulous in how you make your beer, but it's easy enough to just take a beer that you've got and pre-dry hop, pull some of that beer aside, and let it ferment out completely, and then dry hop the rest of the beer and measure the difference between the two, and see that you'll see that the non-dry hop beer should have a higher final gravity than the dry hop beer, and that the extent of that change is kind of the extent of the hop creep. And so you can do that at home if you've got a shaker table or stir place like that will move things along quicker, because what you're looking for in the dry hop case is for yeast that are present to basically start mopping up any of the fermentables and reducing the gravity. So in the absence of that, you just get a mixture of dextrins and fermentables. So that's quite an easy way, and that's, I think, what many brewers do. They may not use a hydrometer and measure apparent gravity, but they might use, say, an alkalizer that gives them alcohol concentration and apparent gravity, so they can look at changes in ABV or changes in fermentability or changes in final gravity as well.
Mm-hmm. Or changes in real extract too, right? The actual real extract change, not just the alcohol's influence.
Yeah.
Mm-hmm. And RDF.
For a brewer that is concerned about it in their beer, it makes sense for them to do this measurement in their beer. And because the mashing regime makes, if they're making a wort that is inherently high fermentability, they're probably not going to see dry hop creep a lot. They could look at, test batches of hops that are coming in. But if the fermentability is high, and when I say fermentability, I'm talking like an RDF that's above 70% or 75%, then they are probably not going to see it in as much as if the beer had a fermentability of 65% or 60%, which is pretty low fermentability. And I'm talking about real degrees of fermentation, not apparent degrees of fermentation. And so using that beer matrix as the medium to test hops is a perfect, easy way to do it. So you got that beer, you're constantly making in your brewery. You pull samples from that pre-dry hopping, and then add a known amount. We typically add 10 grams per liter. It's like, not quite 2.5 pounds per barrel. That's a decent amount of hops to see an effect, and let it creep. And we put ours in an incubator and warm it up to 30 degrees Celsius, so like 80 something Fahrenheit. But you can just leave it on the bench, and it'll go a little bit slower, but it will still occur. And if you could have it on a stir plate, it will go even quicker. And you can then see the effect of a particular hop lot that you're trying to compare. So that's kind of the easy approach. Not easy, but it's a relatively straightforward approach. The sort of challenge comes like when, like if a hop supplier wants to create a spec that says, okay, this is what the enzyme activity of these hops are. Like, how do we do that? And so there's one approach is to look at enzyme kits. So there are companies like Megazyme that make enzyme kits and specialized substrates to measure things like the diastatic power of malt or the glucan content of malt. They use, either they're testing the enzyme activity or they're testing the enzymes with specialized substrates. And we use some of those stuff when Cade's work. And it's a little bit of a challenge because the enzyme levels are so low in hops. Another approach is to use a standard beer or a standard starch substrate. The hop or the barley industry has done that for years to measure alpha amylase. They take a soluble starch solution that you can buy and it's sort of tested and calibrated, and the malting companies will use that to understand what the alpha amylase activity is. And we started down that road a little bit ourselves. We started looking at like maltodextrins as a potential dextrin source. And it was kind of unsatisfying. It didn't seem to match what we saw in beer. But then again, we looked at, I think, 6 or 7 different commercial beers and that were undry hopped, and we dry hopped them, and they all had different responses because of how, like, the wort composition. So that becomes kind of complicated. But if we could go down that route of having a standardized starch solution, that would probably be ideal. There's some work that the guys in them at TU Berlin, Philip Beachstock, and Brian Gibson have done in that area, looking at trying to create a standardized starch solution. They did some interesting, like, design of experiments approach to look at different levels of carbohydrates, or not carbohydrates, but of starch, different temperatures, different times, to try to dial in like a standardized approach.
Yeah, looking at different sources, because I mean that is one of the methods that or the main method that we use is the beer, right? Like using a beer.
Yeah, we use beer plus an HPLC. That's the other thing.
Exactly.
Is that we have a expensive equipment on the back end of it. So rather than using yeast to tell us how much sugar was there and have them by measuring a reduction in gravity or increase in alcohol, we actually put something in to prevent any microbiota effects. We put a really powerful antimicrobial, and then we measure the changes in sugar composition with the HPLC, which I like because now we're measuring specific individual sugars, and it's an expensive approach. But if you have a lab that has an HPLC, then it's just like it's a different kind of instrument.
Yeah. It's just measuring it. Yeah, if you've got an HPLC already set up, it's just measuring a dry hopped beer. Right?
Yeah, exactly. Mm-hmm. Yeah, it's just get a little bit of expensive column, but the reagents aren't that bad, and because you do this all the time, Cade, and you have a sense on how long it takes, but it gives you a really nice set of numbers because you know exactly how much sugar is being produced from the substrate over a given period of time.
Yeah, exactly. And of course, in our analysis too, we're also always measuring the base beer as well, because assuming that there are going to be changes.
And this is like kind of a classic approach to biochemical assays, are these time point assays where you incubate something with an enzyme for a period of time and then look at how fast it was working.
Mm-hmm. Yep. Exactly. Which is so that's kind of like again to measure and see what you're doing with hop creep is start like that easy way, like just observe the differences in attenuation via hydrometer and or whatever you're looking at, alkalizer, and then gets more complicated into the HPLC, and then of course you can even do proteomic work, which I think Brian and Philip are looking at in at the over there in Berlin, which is pretty cool. I hope to once they've published a preliminary study, which I should get them on and talk about. But once they sort of finalize their methodology, it'd be fun to get them on and talk about that too. So I now want to transition over to factors that influence hop creep, knowing that we don't have a lot of time left on the episode to talk about it. We have already discussed quite a few things. We discussed, variety, how variety plays a role, differences in variety. We spent a lot of time talking about that in the prior segment. Of course, you heard what I've looked at in terms of terroir and maturity in my defense. But there were, and of course variety. But there was one the study that sort of kicked off the hop creep research with your lab, Tom, was the Kaylin Kirkpatrick's work back in 2018, and that was a really cool set of studies where you looked at not only differences in varieties, which we've mentioned, and not only developing an assay to measure hop creep potential, but also what are the tools, what things that brewers do impact those. And I'm thinking primarily about like time on hops, temperature of the dry hopping, dosage, the amount of hops that you add. Do those things matter to hop creep?
Mm-hmm. Yeah. Thinking back to like her work, and we were talking about just in the previous little discussion around how do you measure hop creep, and are us trying to find like a soluble starch that seemed to be like the ideal for me, because then other labs could use it. But after playing around in that space for a little bit, we realized it just wasn't, like I said, very satisfying. The results weren't matching what we were finding in the beer. And so we made this decision, let's just stick with beer, right? And we looked at, like I said, 6 or 7 different beers. We found one that had high residual dextrins, that wasn't dry hopped, and was close. So this was like Ninkasi Total Dom. And so, okay, we'll just do work with one beer and kind of have that comparison. And then the next project came, well, we probably should use that same beer to make some comparisons. And here we are, find ourselves 7 years later. We're still like, call up the guys and then say, hey, can we get some more so Total Dom that we can do the work with? And then we got the call or the response this year, which was, oh, by the way, guys, Total Dom, as you know, is no longer. It's now going to be dry hop. So again, that was like the wah wah wah.
Well, it's okay. Yeah, we still the good thing is a guy at Ninkasi, one of your former students, Dan. So he was able to hook us up, and we're able to still make that beer, which is great.
It's just it is presenting like all these research projects that we are publishing are for the most part somewhat comparable because they're using the same beer. Now it's every year it's clearly a different batch of beer. We haven't just made this one giant batch back in 2018. We're still doing the analyses. Pretty much every harvest year we change over the beer, but all the work that's done for that harvest year is done with the same batch of beer because we don't need that much beer. We probably have 2 kegs of this or maybe 3 that we go through.
Yeah.
Mm-hmm. So it's like it would be nice to see, Philip, if we can get a starch substrate that mirrors to some degree what's going on in the brewery, then that'll be good. But to answer your question, like what kind of things did we look at, and that influences that the different like processing, like brewing processing systems had, were things like hop dosing rate. And so it's not unsurprising that as you increase the amount of hops that are being used in the dry hopping process, you see a larger and larger effect. Now, these enzymes are catalytic in terms of the effect that they have, but we're not at a point where the system is like saturated in the enzyme. We have excess substrate, and we have really, really small amounts of enzyme. And so as we add more enzyme in the form of more hops, we see a response. Like in many cases for enzyme tests, when you're trying to get a sense on enzyme activity, you don't want to have this to be the case. You want to have we're going to have sufficient enzyme that your reactions are not dependent upon substrate concentration. So
There's kind of a time component to that too, right?
yeah,
Because
exactly. So the hops, the enzymes seem to be somewhat associated with the plant material. Um, and that is that the longer the time that the hops were in contact with the beer, the greater the impact that we saw. So like if you dry hop for 1 day versus dry hop for 7 days, and what we would do is we would dry hop for 1 day and dry hop for 7 days, and both beers would be tested like on day 8. So it's not like we dry hop for 1 day and then we test it. Like we dry hop for 1 day, take that off the beer off the yeast, we'd filter it and then measure it at day 8. And then we do the same thing with the beers that were dry hopped like 7 days, and then filter them the next day, measure them. And we would see less of an effect like this. If the beer was in contact with the hops for only a day, we would see less hop creep than if they were in contact with the beer for 8 days. But so some of the enzyme activity is occurring that enzymes are leaking into the beer and they continue. And we talked about that in terms of like with packaged beer, right? The beer has not been in contact with hops for months, but it's still producing more and more sugars because these are packaged, unpasteurized, but filtered beers. So that some of the enzymes are leaking into the hops. I mean, some of the enzymes from the hops are leaking into the beer. But I would say the bulk of it are attached to the green part of the plant of the cone itself. And so longer time on hops result in greater impact. Hop products that have less green matter, like concentrated hop products, like a T45 or a T35 product, has less of an effect. It's not zero. It's maybe half. I mean, we talked about kind of these thresholds. So it will definitely have an impact. Like if you're a brewer and you're looking to try to minimize this, you could, by moving towards T45s for at least part of your hop build, that will reduce the amount of impact. Dry hopping temperature has an impact. So if you dry hop at warmer temperatures versus colder temperatures, again, not unintuitive, right? Because these are enzyme driven reactions. There's enzyme kinetics. And so at higher temperatures, you get greater activity than you do at lower temperatures. Moving like to complete downstream products would prevent this in its entirety. Like if you were to or even just like a mixture of some hop material with some advanced like hop aromatic products, then you're going to really reduce the amount of green material that goes into the tank that's being dry hopped. It helps in terms of brewing yield, but it will also help in terms of the extent of the hop creep activity or how much change and attenuation you would see.
Mm-hmm. Yeah. And temperature again makes a lot of sense, right? Because like you said, it's an enzymatic process. So it's warmer temperatures, the enzymes are more active. And
the challenge is that if you were to do, let's say, a 7-day dry hopping warm and compare it to a 1-day dry hopping cold, well, forget that. How about just like 1-day warm versus 1-day cold? They're different phenomena. And that's kind of the problem, is that when you talk to brewers, like the quality of the hop extraction that they get under warm conditions versus cold conditions is different. And so that's part of like many brewers like to dry hop warm because they feel like they get greater aromatic extraction than if they do cold. And so it's like a cold short contact time is a potential challenge, but there are so many different ways to dry hop beer. I can see, if you filter beer to remove yeast and you use an agitated dry hopping system cold, so you get really good mixing, and then a centrifuge to remove the particles, the actual extent of the hop creep is not going to be huge. And then you package the beer filtered or centrifuge so that the amount of yeast that's going into package is extremely low compared to warmer temperatures, kind of like old school, dump the hops in, just let them sit, keep it warm.
Yeah. So from a brewer perspective, right? I mean, that's some key takeaways there for them is like, okay, yeah, the temperature can play a role in hop creep, the dosage, certainly the amount of hops that you add, the time that you have the hops on the yeast.
The amount of yeast in suspension.
Yeah, the amount of yeast in suspension. Yeah.
So that's brewers who like when they're battling this, like depends upon if they're trying to work with it or prevent it. Because you can work with it, right? You could just say, okay, we know it's going to happen. Like kind of we talked about the brewer who was surprised that we would ever have like an apparent gravity above 3. So if you're dry hopping towards the end of an active fermentation, you have a non-flocculent yeast. You got a lot of yeast in suspension, and you got a long time on the hops. Then the hop creep is going to occur, and the beers they'll certainly dry out. They're not. It's not like you're making a brut IPA by going this route, but you will certainly dry the beers out more than if you hadn't dry hopped. In comparison to someone who really doesn't want the beer characteristics, that is the fermentability, residual extracts to change. In that case, to try to, well, in the former case, you're kind of working with this and in trying to make sure that hop creep is happening in the cellar and everything's kind of done changing before the beer goes to package. And another approach is to kind of work against it, centrifuge out yeast, lower temperatures, short contact time, and then filtration or centrifugation to make sure that there's very little material plant material that follows into the finished beer, as well as yeast. And in that case, you can kind of work with having a decent hop aromatic extraction and a minimal impact that the hopping process has on the beer.
Yeah.
And I think there's some, I guess it's pros and cons. Right? People think about biotransformation of hop flavors in the presence of yeast. And it does occur. There's a lot of scalping that goes on as well. Like the yeast is just taking out a lot of hop aroma. And so dry hopping in low yeast conditions or the absence of yeast, like in a large brew, like if you take beer at the end of fermentation and run it through a centrifuge, so it's basically bright, not bright, but yeast free, and then do the dry hopping and do it cold. Or you could even do it warm, but you're gonna have minimal amount of re-fermentation in that case. Can be an approach.
Yeah. I mean, so lots of tools and things that a brewer can use there. Lots of tools and tricks and tips for a brewer to use to avoid or mitigate a hop creep. But before we left the subject of hop creep, I wanted to mention, of course, Lindsay's work recently looking at kilning. We teased that a couple of times earlier. We already talked about how enzymes may be a little, temperature sensitive, meaning like the temperature of dry hopping, but at high kilning temperatures, you're up near that denaturation point, or at least like a significant reduction.
Yeah, that was one of the things that we kind of discovered, not on accident, but it was something that we really were thinking about on the front end with Lindsay's work. And she did 5 years of kilning work, and it was really trying to understand the degree to which kilning temperature has an influence on hop quality. And what we found is that hops are surprisingly insensitive to kiln temperature. You can definitely screw up hops by kilning them too high, as well as too low. Particularly if you don't have sufficient airflow going through the bed. The hops, like if you have relatively cool hops going into a kiln and you've got insufficient airflow and you've got a fairly high air on temperature, the moisture that comes off the kiln, the bottom part of the kiln will just condense on the top part of the kiln. You actually start sweating the kiln mass. It's kind of like you're stewing, cooking the hops, which is not good. But if you have enough air to kind of make sure that you don't cause that phenomenon to occur, the temperature didn't seem to have a big impact on the hop chemistry or the hop sensory. It was really quite modest. What the temperature did have a noticeable impact on was its hop creep potential, and not unsurprisingly, as we went to higher killing temperatures, the hop creep potential went down as we start denaturing these enzymes in the hops. So that was kind of a neat output from that work. It was okay, higher kiln temperatures could result in lower hop creep, quicker time through the kiln, and then for some varieties, lower energy usage because the dwell time in the kiln is so much shorter. So there were all these kind of positive things that came out of potential positive things in terms of kiln temperature that were really counterintuitive. People thought, okay, killing at higher temperatures, you're going to lose aroma and it's going to cost more energy, all this stuff. And there was all these, everywhere we turned, there were wins. It was so.
Yeah, and I mean, another one last piece of that spawned the maturity project, right?
Yeah, that's right. And that's kind of full circle back to your project, which was basically when Lindsay was doing these projects. She would do on-farm reps, and it was just curious that all the rep twos were either the same level of hop enzyme activity or considerably lower. Some of these reps we would do a day or two after the first rep, oftentimes like one day. But there were some instances where, because of time and whatnot, we just basically left the farm for a week, and we'd come back, and so we're kind of measuring hop creep at the front end of the harvest, and then we are doing this killing trial at the front end of harvest, and then we do it at the back end of harvest, and notice a really big change. And so that kind of got the question of going like, man, does hop maturity have an impact on hop creep? And Jason crawled and said, yeah, let's do this trial at our farm. We'll just like set you up at the field, and we'll just harvest it every week for like 7 weeks. That's like what you started working on when you came in the lab, was like all these piles of hops that we got from the folks at the farm.
Exactly. And now we're going into year 4 of
I
that
know.
study.
It's pretty amazing. Year after year after year, like that influence is so consistent that as you and the output is that as the hops mature on the vine, the hop creep potential goes down.
Yeah, exactly. Yeah. But again, coming from what Lindsay was seeing on the kilning trials, it's just so interesting. I mean, I started the show asking like how you keep interested in hop questions. And I mean, this is how, right? It's in
some cases, the research just spawns new questions that are open to seeing these differences. Oh, okay, let's look at that some more. In other cases, you just kind of close the book and then, okay, what's new and interesting? And, like your work right now that you're in year 2 on wildfire smoke, that's like, that was sort of propagated by the big wildfires we had in 2020. And then again in 2021, 2020 here in Oregon, it also affected Washington, but 2021, when it's like 6 weeks of smoke in Yakima Valley and these large recalls of contaminated hops, or this like defective hops. And then again in 2022, when we were setting up at Elk Mountain, that huge fire there. So 3 years of fires in each of the 3 hop growing regions was like, hey, we need to get our hands around this problem. So that's what's steering that project, is like the presence of wildfires. And I guess we'll see. This year it's been pretty decent here in Oregon. It's been so in Yakima. I think it's been fine in Idaho. So
yeah, we'll see. I mean, certainly something to look forward to with smoke. And then, of course, you've got Cecile's aging project and all of those other things that are going on in the lab. So we'll have a lot to talk about.
Yeah, we will. Exactly. We need to come back before episode 340, right?
Exactly, right? Exactly. Although, who knows? I'll probably still be in the lab at that time getting my PhD.
No, you won't. You're moving along just fine.
Yeah. Well, I as a wrap-up question, I always ask this of my guests at the end of the show, and if you wanted brewers to take away just one thing about hop creep, what would it be?
Wow. Okay, that's a great question. Oh my gosh, yeah, you can kind of stump me. One thing. Yeah, I think that the one thing is that it's going to happen every time you dry hop beer. You're going to have some hop creep phenomena. It's just the combination of the type of beer you're making and the hop bills that you're using will influence the degree to which it's really a phenomenon for you. It may be just because of the hopping regime and the timing and the particular hops you use. It may be a non-issue. It doesn't mean it's not happening, but it's just happening at a low enough level to not be an issue. But for some of you, it's going to be, or is, it causes a profound change in terms of the fermentability, residual extract, alcohol concentration, dryness of the beer.
Yeah, you're going to see variation depending on your brewing processes, right? Yeah. The hops that you use in your brewing process, you're going to see variation. But thankfully, there's a lot of research out there now to look at, and a lot of that has come from your lab, Tom. So,
cool. What a journey we've been on. So, and that we're continuing on. We're not getting out of the river yet. We're still like,
oh no, we're still looking at this. Yeah, we're still looking at how even the smoke project will potentially have an enzyme component to it. At least we'll look into it. I don't know if there will be anything to it, but we'll look at it.
You never know, right?
Yeah. Exactly. You never know. All right. Well, thanks again for coming on and talking about hop creep. This was fun. We've got to get you back more frequently than every 165 episodes.
Yeah, exactly. Maybe we do this every 6 months or so, and we kind of like, what's new in the lab?
Yeah, what's new, or what are we doing? What's the next research project that's going on? That'll be fun. Well, thank you, Tom, for joining me in the Brew Lab.
Yeah. Thanks, Cade. Appreciate being on this show with you again. All
right. Well, next week, Jordan and I will be back applying the science of this episode. See y'all then. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.
At Grand Canyon University, we believe in equal opportunity, and the American dream starts with purpose. Whether your pursuit involves a bachelor's, master's, or doctoral degree, GCU's learning environments are designed for supportive networking and collaboration. With over 330 academic programs, GCU provides a path to help you fulfill your dreams. The pursuit to serve others is yours. Find your purpose at GCU. Private, Christian, affordable. Visit gcu.edu.